Hello everyone. In this lecture, we learn about attenuated vaccine, their characteristics, how we can develop an attenuated vaccine, their advantages and disadvantages. Okay. So, what is vaccine? We all know vaccine means the preparation of antigenic material which is used to induce active immunity against pathogenic organism. That means we take vaccine to get immunity against that organism. Okay, so there are basic two types of vaccines: subunit vaccine and wholesale vaccine. And wholesale vaccine can be killed vaccine or live attenuated vaccine. Okay, so in this chapter, our lecture is focused on live attenuated vaccine. Okay, so the live attenuated vaccines, they contain non-pathogenic relatives or attenuated strains of the pathogen. Okay, which are almost or entirely devoid of pathogenicity but are capable of inducing a protective immune response. Okay, so that means the live attenuated, the microorganisms that we use as live attenuated vaccines, they don't have the pathogenicity. Okay, that means they cannot create disease in their host, but they can induce the immunity. Okay, so and one good part of using live attenuated vaccine is as they are live, they can multiply in the human host and provide continuous antigenic stimulation over a period of time. Okay, so now which pathogens we can use as, or which microorganisms we can use as live attenuated vaccine. That means the candidates of the live attenuated vaccine. So we can use relatives of the target virus, okay, which are virulent for another species but avirulent in humans. For example, for a long time, cowpox virus was used as a vaccine to protect human from smallpox. Okay, cowpox is a relative of smallpox, right? They are uh, smallpox. Okay, so cowpox, they cannot cause disease in the human, but they can induce immunity in the human, and this induced immunity can protect the human against a smallpox infection. Okay, this so relative of the target virus can be used as a candidate for live vaccine. Okay, also we can use virulent strains which have been attenuated naturally or by classical methods or by recombinant DNA technology. That means we can use a attenuated virulent strains as a vaccine. Okay, and that attenuation can be done naturally or by using some classical approach or the modern recombinant DNA technology. Okay. Then another candidate for live attenuated vaccine is microbial vectors containing foreign genes that encode a vaccine immunogen. For example, HBs-AZ, hepatitis B surface antigen vaccine. Okay, it was developed by using an attenuated vaccinia virus carrier. Okay, that means the antigen, the gene of HBsAg was inserted into a attenuated vaccinia virus, and this act as a vaccine for the HBsAg. Okay, hepatitis B. So, in this type of vaccine, we don't use the whole. For example, why we take the HBsAg vaccine to get protection against hepatitis B, but we will not use the whole hepatitis B virus as a vaccine. Instead, part of the gene which encode the surface antigen of hepatitis virus was inserted into another virus, like attenuated vaccinia virus. This attenuated vaccinia virus is called a carrier because it carries the antigen of the, it carries the gene that encode the surface antigen of HB. So this recombinant virus can be used as a vaccine. Okay. So now the ideal live attenuated virus vaccine, they should possess some characteristics. Characteristics means they should do something and at the same time they should not do something. Okay, so like the attenuated organism should mimic the natural behavior of the original microbe without causing significant disease. I mean, it will behave like the wild virus. I mean, wild pathogen means like the natural pathogen, but they will not just cause the disease. Okay, and the attenuated organism should not revert to virulent by any factor, say such as you have a virulent I mean attenuated vaccine. Okay, a attenuated organism. So you want to use it as vaccine. So you vaccinated a person with that vaccine. But if somehow this attenuated organism it revert, that means it gain its virulence, then it will cause disease to the host. So we cannot use such kind of attenuated organism as vaccine. Okay, the attenuated organism should be sufficiently invasive and persistent to stimulate both a strong primary and lasting memory immune response. It means the vaccine, the organism, after it, after we vaccinate it into a host, it should remain in the host body, right? Otherwise, it cannot induce a long-lasting memory immune response. Okay. Then the vaccine should not lead to the development of a persistent carrier state. Okay, we know. Sometimes the microbes, they persist in our body, and we become carrier of those microbes. So a vaccine, they should not they should not persistently, reside in our body. Okay, so that means we should not become carrier of the vaccine organism. Okay. The vaccine should minimize tissue damage that is not needed to induce an effective immune response. Okay, some vaccine they can, some live attenuated vaccine they can cause some tissue damage, but those tissue damage should be essential for immune to inducing the immune response. Okay, so that means a good attenuated vaccine, live attenuated vaccine, should not cause disease to any tissue which is not necessary to induce immune response. And then the vaccine should induce immune response largely at the site of the natural infection. So okay. So natural infections means, we know different organisms, pathogenic organisms, they cause infection in different organ or tissues. Okay, so it is desirable that the candidate vaccine will induce the immunity in its natural area of infection. So such as if it infects the lung, it should induce immunity in the lung. Okay, otherwise it will not help us, right? um, then the vaccine should raise immune response to all protective antigens of the desired pathogen. A pathogen don't contain only one antigen, right? It contains several antigens which protect it. Okay, it's protected from host immune system, right? Like if a pathogen want to cause disease to its host, it has to escape the host defense mechanism, right? So for this, the pathogen they use different kind of virulent factors. Okay, so we call this antigen. So a good live attenuated vaccine should induce immunity against all these virulent factors. That means against all these antigens of that organism. Then the vaccine should possess some containment features to reduce its shedding or survival in the nature to preclude vaccination of individuals who did not elect to vaccinate. I mean, once the vaccine candidate is in the environment, they should not survive there. Then, vaccines should have a reasonably long shelf life under the usual storage condition. If a vaccine have a very short shelf life, we cannot store it, right, for a long time. So a good vaccine shelf life of a good vaccine should be longer, and it will be better if we can store the vaccine in like room temperature. But in most of the time, we actually cannot uh, store live vaccine in the room temperature. We usually have to store them at cold temperature. Okay, and then any live bacterial vaccine should be susceptible to all clinically useful antibiotics. It is important because these are live organism, right? So it can happen. I mean, in some host, the live, I mean, although they are attenuated, but in some host, still they can cause disease or infection. So if this if they cause the infection, then uh, eh, we have to use the antibiotics, right? So if it is a bacteria, we have to use the antibiotics. If it is a virus, we have to use the antiviral drugs. So the vaccine should be susceptible to all kind of clinically useful antibiotics. Okay, so if the vaccine itself have drug resistance, then in those kind of cases, I mean in accidental case where the vaccine will cause infection, we cannot treat them if those vaccines are itself drug resistant. Okay, so these were some required characteristics of an ideal live attenuated vaccine. So now I'll talk about the approach or process by which we can develop a live attenuated vaccine. Okay, so there are two types of approach: classical approach and modern genetic engineering approach. Okay, so I'll discuss about this approach in the next video.